Good morning, fellow humanists. My name is Todd Kimball. It's an honor to be your MC this morning. Happy first day of fall. That humanism is a rational philosophy informed by science, inspired by art, and motivated by compassion. It advocates the extension of participatory democracy and the expansion of an open society standing for human rights and social justice. This morning's presentation will be a brief history of the scientific discoveries enabling gene replacement and gene editing to become useful and potentially transformative therapies. These innovations come with profound ethical considerations. We have a true leader in the field to guide our discussion this morning, Dr. Rafael Escandon. Rafael has worked in global clinical research and development and patient advocacy in the biotech industry for nearly 30 years. He is currently the founder and principal of DGBI Clinical Research and Ethics Consulting, a consultancy to patient advocacy groups and to the biotech industry. His approach is to be highly considerate of the ethical implications of conducting and participating in clinical research, especially in rare diseases and in vulnerable and developing world populations. He is currently a member of the Pediatric Gene Therapy Medical Ethics Working Group in the Department of Medical Ethics at the NYU School of Medicine, as well as being adjunct faculty in Kansas City and at UC Berkeley. Rafael holds graduate degrees in cardiovascular physiology, public health, bioethics, and is a certified healthcare ethics consultants. For Rafael Escandon.
I'm almost speechless, but I did promise my younger brother, and hopefully, this is permissible. I've been texting with him since we got the tech set up this morning, and told him what I was doing, and he did a little research on the Bay Area humanists and the Portland area humanists, and he said, please open with a joke. So I'm going to open with this joke that he just wrote to me, which says, when most people ask me to discuss the ethics of gene therapy, all they want to know is who the hell Gene is and why I should listen to him. So he didn't use the word hell, by the way. So I don't know if that, I thought I'd turn that down a little bit, but I'm really thrilled to be here. And just the connection is, actually, I lived in the San Francisco Bay Area for almost 25 years and worked with the daughter of Deborah, who's part of the Bay Area humanists. And I've spoken to them a couple of times on the ethics of research, and then given a very similar presentation to this one just a month or so ago. So I'm really looking forward to meeting all of you, interacting with all of you. If we can, we'll hold questions to the end, because I think there's like a nice organic nature to how questions build on each other. And what I'm really going to try to do today, though, is there's a lot of history here, I think, that's important in terms of, I think it's both from a curiosity perspective, but also it lays a little bit of the technical foundation that's necessary to understand a little bit of what we're doing and what's being done in the gene therapy approach to the treatment of various diseases. And so that's really my area. But the idea of the ethics of genetics in general and the harnessing of that knowledge that's come from the Human Genome Project, that's come from the application of various technologies into medicine and informatics is enormous. And I think we will continue to grapple with some of the intersections between the broadest technical use of that information in the most responsible way, and the ways that how our society and our norms and our ethics can sort of put guardrails around those and harness the misuse and overuse of that information and those technical elements. So with that little background, I'm going to share my screen here and hopefully get this working just right. So I'm going to run you through some history. I'll do a good quick disclosure here. I'm not going to read everywhere on the slide. I do consult for a few companies on the in the biotech industry, mostly to do safety and clinical trial design type functions. I don't own any stock. I'm a board member for a rare disease foundation called the Cure GM1 Foundation, and then as you heard Todd say, I'm a working group member for both the PGTME at the NYU Grossman School of Medicine and also for the American Society of Gene and Cell Therapy. I'm on one of their committees for the development of genetic and metabolic gene therapies, and that's a professional society that focuses on the clinical applications of gene therapies. So let's just set the background here. What is gene therapy? What does it mean? It's actually, from an ethicist perspective, it's unfortunately, unfortunately named. The name, it's an interesting story to really even trace back the name gene therapy. Therapy, from an ethical perspective, implies that whatever intervention it is going to benefit the patient. When something's in the research stage, however, those therapeutic benefits have not been proven yet. So what do you do when you're researching a gene therapy? Is it a potential gene therapy? So these are some questions we grapple with on a daily basis when we're doing gene therapy research. But the idea really is, it's a sort of bottom caption here, is that gene therapy is a promising approach to address the underlying genetic causes of diseases with the ultimate goal of improving patient outcomes and quality of life. And so there are a lot of different ways to do that. There are a lot of potential applications. But the way we started with gene therapies in humans and with these interventions was to look at the sort of catalog of what are called Mendelian diseases, and I'll go into what that means in a moment. But really, to really crystallize that, or to boil that down, Mendelian diseases are diseases that are related to a mutation in a single gene, and so the whole disease process and all of the negative elements of a particular disease, a genetic disease that's a Mendelian disease, are related to the fact that one single gene isn't producing the protein that it needs to produce a functional version of that protein. And I'll go into a little bit more about what that means in a moment. Here, this is just a presage. We're not going to get to all this, but this is just a sort of menu of the ethical considerations that I deal with every day, working with my biotech clients and working in academia, when we think about what the implications are of doing research and using these therapies, in the commercial medical setting. You've got issues of safety and inheritance concerns when you modify a gene in a human. When you've given them a gene therapy, what does that mean 5, 10, 20 years down the line? There are currently guardrails around manipulating genes that will actually continue to be inherited down the population tree, which again we could give a whole other talk on heritable gene editing, but we won't go really into that today. But issues of privacy and informed consent. When you voluntarily give your genetic code to a company like 23andMe or Ancestry.com, what are you really giving, and how are they using it? And do you have the time or the interest in understanding the hundreds of ways they're going to use that and maybe export your genetic information? So those are real concerns that we have about the use of gene therapy and genetic information, and also How do you properly inform someone about the myriad ways that your genetic information could be used and distributed? For the therapies that I'll talk about, they're extraordinarily expensive, and so unfortunately available to only a few, and depending on the healthcare payer arrangement in whatever country you live in, the availability of a gene therapy as a treatment for a condition may or may not be available. It's pretty much not available anywhere in the developing world, and really fairly, it's still pretty new in the developed world. Who regulates and oversees this? I'll talk a little bit about that. We'll talk a little bit about the ethics and the quandary around whether we should or shouldn't do things like enhance genes. That is sort of to, have them endow them with maybe some superpowers that would prevent things like aging. That's a really enormous, complex area of research now, but there's an enormous amount of social interest in that. And then back in the treatment element of gene therapies, we're really grappling with this fact that you can't currently re-administer a gene therapy. And I'll talk a little bit about that because of the way that that the gene therapies are delivered. So that's just a menu of things that I hope we can get to, but I wanted to put that up front so you can be thinking about some of these questions with some of the information that I go over. So who do we have to thank for all this? Let's take everybody back to high school biology and maybe undergraduate. If you ever took a genetics course, Gregor Mendel is considered the father of genetics. He was a very patient person, but he did have some time on his hands. So he was a monk. Turned out to be a monk. He actually wanted to be a teacher, but didn't. I guess the teacher's exams were pretty difficult, and he didn't do super well on those. So he ended up going into a monastery in what's now around Brno in the Czech Republic, in the Moravian portion of the Czech Republic, and he was, like I said, very observant and an avid gardener. Had a real green thumb, and so he made observations over generations and generations and generations and generations of pea plants, of wild pea plants. And so there are several characteristics, I won't go into all of them, but he crossed them, different colors, different shapes, different bloom colors, different pod colors, different pod textures, all sorts of different combinations, and mapped out the inheritance characteristics of all of those crossings of pea plants, and actually noticed very specific patterns in the inheritance of these certain traits. And through mapping those out and documenting them very carefully, his observations on inheritance characteristics, what we've come to know now as dominant and recessive characteristics for single gene related phenotypes or presentation types. Mendel is the person who first laid that out, and he did that in 1856. He summarized all these experiments, and his notebooks are really something to see. And then, of course, all that rigorous work was lost for 35 years. And so it was then rediscovered around the turn of the last, 2 centuries ago. But some, scientific investigation had, science didn't slow down completely. But it really wasn't, if you go just fast forward to the bottom right of this slide, it wasn't until about almost 100 years later that in 1953, when James Watson and Francis Crick published their very famous work on the double helix structure of DNA and really defined DNA as the genetic material. And so I'll go into that a little bit, but you can just see that that it took about a century for Mendel's observations to crystallize into sort of a molecular understanding of the biology of inheritance, if you will. I won't go into every one of these elements here, but this is just I love this slide because it just shows you how meticulous Mendel had to be when he was doing these crossings of tall and short plants and observing, oh, I get a medium-sized plant after that, and then what happens if I do, a different so that my prediction is this, and then what happens if I do that? Okay, I get 3 talls and 1 short. So what does that mean? And just to go through his analyses of those kind of characteristics on seed color, pod form, pod color, flower, blah 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 blah. It was just really a pretty. I don't have the patience for that. I don't know if anyone in the room or on the Zoom call does, but real analog type of work that he had time to do, but it really has turned out to be critical in understanding how certain characteristics are inherited. And when there is a genetic mutation, it follows quite often, 9 times out of 10 follows a Mendelian inheritance pattern. Real quickly here, just to finish off a little bit of the history and a little bit of the structure. Again, fast forward to Cambridge, James Watson, who's American, Francis Crick, who's British, really published the seminal paper that included work and unfortunately was not properly acknowledged by Rosalind Franklin, who actually did the famous X-ray crystallograph of that showed us that DNA indeed was in this very now well-known double helix orientation that is critical to its unraveling and to the transcription of mRNAs, which are the next shuttling stop in producing actual proteins. So the DNA within the DNA double helix are these individual nucleotides that in triplets of 3. Now I'm going to this next slide here, and it's really to the far right panel, the genetic code panel. These particular individual nucleotides are read in triplets, and each triplet codes for an individual peptide or an individual component of a protein that ultimately gets strung together. These amino acids like serine, tyrosine, asparagine all get strung together like pearls on a necklace into a 3-dimensional protein. So it's often hundreds or thousands of these individual amino acids that create a protein that functions. So that's the whole, that took 100 years to figure out from Mendel's individual experiences with those pea plants. Quickly though, I want to just sort of throw down what that enabled. And so I love this quote from Francis Collins, who's head of the NIH for a long time. And he said this back in, I think it was 1991, that gene therapy has the potential to revolutionize the way we treat many genetic and acquired diseases. And so, as I mentioned earlier, you've got, any number. We've got about 10,000 diseases that we know about that follow Mendelian characteristics that have Mendelian inheritance. So there's a mutation that is passed down in a Mendelian way. And so the Human Genome Project, which you can see on the sort of right side of this diagram, helped us to understand, okay, we've sequenced all the DNA in the entire human genome. What gene is associated, what portion of that huge map is mutated when a certain disease is present. And some of these diseases are very rare, but that was the first step, is is to have the Human Genome Project just understand the entire, A to Z map of our genomes. So, in the center of this diagram, once you've identified a gene that is that's responsible for a specific disease, by cloning it, you can do a few things. Down the middle, you can develop a gene therapy. I'll talk about that. But it also does a couple of other things on the right and the left of this diagram. So it's not mapping the human genome and cloning a particular gene isn't just specifically related to developing a specific gene therapy, but it helps with diagnosis. It helps with putting together preventive medicine strategies and understanding that certain populations of people have, a genetic footprint that makes them, for example, a little bit more sensitive to the effects of a certain drug, or insensitive to the effects of a certain drug, which can make dosages an issue, or allergy, or just a whole universe of elements over on that side. And then you go on the right side of the diagram here, and if you're able to, thanks to the Human Genome Project, understand the normal protein orientation. So say most proteins are enzymes, and most, the proteins that are produced by the transcription of genes, have many functions. But if you can understand really how a protein works and what it looks like in 3 dimensions, if you want to turn that up or turn that down, you can actually, through that understanding, get some organic chemists or some biochemists involved and actually design some specific drugs, molecules that will change the function of those proteins. So it's not just about a gene therapy or about pharmacogenomics or preventive medicine, but you can also design, get insights into designing specific drugs that will help with various diseases. So that's just and that's just a very small footprint of how the Human Genome Project helps medicine in particular. Another quick timeline here. So as I mentioned, so 1953 was when DNA was determined to be the genetic material, and we understood how it codes for amino acids and then ultimately for proteins. Now, less than 20 years later, the first proposal for gene therapy is a gene as therapy is made by scientists here in the United States, and then 2017, just the timeline is just accelerating. So 2017, the bottom right here, the first gene therapy was approved for use in the United States. Prior to that, though, even about 10 years before, there were other approvals that were done in other countries, whether it was in China or in Europe. And then, in the early 1990s, there was a small successful use of gene therapy. Didn't result in an FDA approval, but it was successful in treating a condition called SCID, which, if you grew up in the 1980s, you remember John Travolta in a like a Hallmark type movie called The Boy in the Plastic Bubble. That's the condition that John Travolta had in that film. And then there was, however, a little bit of a break in 1999. There was a pretty famous death by a young man, 19-year-old named Jesse Gelsinger, who had an immune response to a gene therapy that was given to him, and that really kind of put the brakes on gene therapy to get a better understanding of its safety. But since then, it has just been it has many of those concerns have been worked out. Not all of them, and as an ethicist, I'm very circumspect and cautious about these things, but it has just the modality has just grown enormously after about pretty much a decade of real caution in the United States, at least. So won't go into everything here. Just want to do the first three panels here. Viral vector delivery. That is how most gene therapies are delivered right now. I'll talk specifically about that. Lipid nanoparticle delivery is another way to get these gene therapies into the body. That if anyone got a COVID vaccine, the Moderna and the Pfizer vaccines use these lipid nanoparticles to deliver the mRNA type vaccines that we got if you got those vaccines. And then you've heard a lot about CRISPR-Cas9. That's gene editing. That is gene therapy, but it's a little bit different than what we call whole gene replacement. And I'll go into that a little bit. But again, these methods are constantly changing, constantly improving. We did get the first CRISPR gene editing FDA approval early this year. So that's pretty exciting. Then just one last word about this. As a scientist, I hate this slide actually, but because, the full bright orange circle is not 100%, it's 85%. So it's just, it's a deceiving diagram. But the point here is that when you're designing a gene therapy, and it is so specifically targeted, so specifically designed for the condition, and the protein, and the gene that's meant to affect the production of that protein, that they do tend to be not miss as frequently as a traditional pharmaceutical drug or even a surgical intervention to either correct or modify the problem that you're going for in an improvement type of way. So you don't have as many misses, but sometimes you do, and those are usually not related to the fact that you're hitting the wrong gene. They're usually related to some other technical reason that the gene itself is not getting to where it needs to go, or there are other factors that are causing that protein not to be expressed. But the point here is that it's a pretty precise intervention, and so that is hopefully going to continue to improve. All right, so quick pause. Here we are now at the state of the art. Okay, so this is an example of how the left-hand panel of 2 slides ago that I showed you about. This is an in vivo administration. Imagine you get an IV infusion, or you get if it's a neuro condition, this will get injected into the spinal cord. But if you go across the top panel here, the adeno-associated virus, or AAV as we call it, is it's a virus that's benign to humans. And most people ask, viruses are bad, right? And the ones that make you sick are, for sure, but a virus is over the million or so, or several million years that biological organisms have been, complex biological organisms have been on the planet. Viruses are the best mechanisms to actually get genetic material into an organism. Now, mostly they interject or inject their own viral DNA into that organism and take over your cells, and that's what an infection is. But if you can go across the top panel, if you can engineer out the virus's DNA from that shell that protects it, and introduce the corrected human gene into that virus's outer shell, you've got a ready-made, totally proven system with which to deliver the human gene that was either broken through inheritance or not inherited at all. So, and now if you go to the bottom panel, if you administer trillions of those engineered viral particles, into the body, whether it's through an IV. If you need to get to the brain, quite often it's administered into the spinal fluid in the back, in the spine, or up at the base of the brain. And those trillions of viral particles that contain the corrected human gene, what we call the wild type human gene, the normal human gene, then that's transferred into the nucleus of the target cells, and the proper protein, the functioning protein, gets produced. So that's really how viral-based gene therapy is delivered, and how it works. It needs to get to the cells that produce the protein in the body that is lacking because of the genetic mutation. What's the scale of this? So here's a search I did a few months ago on the big database of clinical trials, called clinicaltrials.gov, and put a few keywords in there, and you can see that there are, as of April or May, about 2,000 research, interventional gene therapy trials. There are, more than half of those are very early stage, what we call phase 1 or 2 gene therapy trials. Of those 1,400, about 500 are currently active, or that is actually recruiting patients, and then you can see the majority of those are sponsored by the biotech industry, versus an academic medical institution like OHSU, the NIH, UCLA, University of Washington. So industry has really jumped on here. So that's just sort of the funnel of what's happening. So big interest in this, in particular over the past 10 years. So here, just going to change gears a little bit. It's a little bit of a complicated slide, but focus on the top panel, because the bottom panel is just a slightly different analysis of this. So this is a slide that illustrates the impact of a gene therapy that's now approved in sickle cell disease. So I want to bring your attention to panel A, the top panel, and the left-hand side, the shaded blue side of the panel. Every row there is a patient. So you see all the red dots on the left-hand side of the screen, in the blue section. Those are all what are a typical and sort of severe condition that happens in patients with sickle cell disease, called a vaso-occlusive event. So just in every one of those individual rows, each one of those rows is an individual patient. And so what you see is that that in the 2 years before the administration of the gene therapy, which is the vertical gray bar, that many of those patients were having quite a few of these severe vaso-occlusive crises, which cause hospitalization. They can cause a stroke. That's really what they are, is they're these mini strokes. But it's an incredibly painful medical emergency when those things happen, and it's very characteristic in sickle cell disease. So the gray vertical bar is when the gene therapy was administered, and then if you look to the right out for about 3 years, you can see that is a transformative effect. No more severe vaso-occlusive events in any of those patients that were treated. So I mean, that's a home run. That's a grand slam home run in baseball parlance. And so that really illustrates to me the potential for transformation of these gene therapies. Now, that's only out to 3 years, and not for everyone, obviously. You've got clusters of patients that don't even have a year's worth of data here, but with the 6-month data cutoff, it's pretty spectacular. So that's I view this as the potential for gene therapy, but how long it will last, I wish we had a time machine. We'll have to see. Similarly, this is another approved gene therapy called Zolgensma in a tragic condition called spinal muscular atrophy type 1. And just a word or two about that condition is it is it's diagnosed in infancy, and patients, these babies die. They rarely see their second birthday, and they have this short development and then rapid deterioration of their motor coordination, and even to the point where like ultimately can't breathe and or even sit up and interact. It's a very difficult, tragic, excuse me, condition. So this is a clinical trial that was done with Zolgensma in babies with SMA type 1. The left-hand panel is the first dose of the AAV gene therapy that was given to these babies, and the vertical scale, the y-axis, is a motor score, just a motor development score, and you can see that in the three patients that were given this gene therapy at this cohort 1 dose, they didn't improve. The dose was raised in the panel on the right, panel B, cohort 2, and you can see again, just from the perspective of that motor score, a transformative effect in everyone except for one patient. And so what was learned here were two things. The first thing is that in panel A, obviously the dose was too low. And then in panel B, it was the right dose. But in the one patient at the bottom that you can see didn't improve, there's also a very narrow window of about 1 to 6 months that you can give this gene therapy for it to have its intended effect. But these are two very positive, very transformative effects of gene therapies that are both approved now in the United States and in the world. So again, the potential for this is enormous. A presentation I just gave at the biggest bioethics conference in the United States just earlier this week really focuses on the type of patients on the left-hand panel, and then the one patient at the bottom. Unfortunately, these patients don't have an opportunity to be redosed with the higher dose because they've received those trillions of viral particles, and so the immune system would recognize another dose. So very unfortunately, and one of the biggest ethical conundrums that we have in gene therapy research is the fact that it can't be readministered. So the typical drug development rubric that companies follow and the FDA asks companies to follow is that you escalate the dose until you find one that's both effective and safe, and then you go from there. But that's all built on a premise of a drug, a small molecule model that those can be turned up, those can be turned down, those can be turned off. Gene therapy can have none of those things. It's a one and done situation. So that creates a real, as a research ethicist, for me, a real conundrum, and identifies a real opportunity to get companies and get regulatory agencies and investigators to think about this a bit differently. So here are just a word cloud of some of the ethical principles in research and how we respect human subjects that are a little bit out of balance with pediatric research and pediatric genetic gene therapy research, when you can only give these interventions once. So what's the justice for those patients who were underdosed, they're not being treated equally, unfortunately, as the ones who have benefited. What is the risk tolerance of parents, particularly who have children who will die with a condition like SMA type 1? They may have a much higher risk tolerance. So if a higher dose has a higher risk of having their son or daughter, say, lose their hearing or go blind, but survive and live an otherwise normal life instead of dying before their second birthday, that might be a risk that they're willing to take. And then pediatric assent is something else that we talk about. Parents, until their children are mature enough to make medical decisions on their own, parents are responsible. But as children reach a certain age before their 18th birthday, we do seek their assent, their active agreement with their parents' medical decisions. And things don't always. There's sometimes differences of opinion. So again, if you're giving something once and it's permanent, it's really more like an organ transplant than it is like taking a pill or getting an IV. So these are all really interesting differences between gene therapies or use of genetic interventions versus traditional drugs or surgeries that we have. And then finally, sort of what's the contribution? How do we acknowledge the contribution of, for example, the 3 patients on the left-hand side of this panel and the 1 patient on the right-hand side of this panel who contributed, very valuable generalizable knowledge. In the case of the patients in the left-hand panel, that the dose was too low. In the case of the patient on the right-hand panel, that the intervention was given too late. How do we acknowledge? How do we reward, memorialize these patients who didn't benefit from the intervention, but really gave us important insights? And I think they're underappreciated. This was the point of my presentation at the Bioethics conference. So just real quickly, the future of gene therapy, it's going to continue to advance. New methods like CRISPR and other CRISPR-like gene edits will continue to be introduced. I hope we will get over this inability to redose these genetic interventions. But time will tell. I wish we had a time machine for so many reasons to go to the future and see, how long these effects last. Are there side effects that are really scary or not really scary that emerge over time with these interventions? It's a very, very there are many open questions that we don't have the answers to right now. But like with AI, this has been going really quickly, and the rules are built on a rubric that's really related to drugs. And so I think this is something that I've been sort of rattling cages about for the past few years, that we need to think about gene therapies and genetic interventions a little differently. So, who regulates these from the therapeutic development perspective? It's certainly the FDA and the European equivalent. There are other organizations like the World Health and ICH, and there are gene therapy and genetic intervention guidelines that exist from various professional groups and research groups, but knowledge still needs to penetrate all these organizations, and practical experience needs to penetrate there too. So we're really not there yet in terms of having a harmonized approach and proper penetration of knowledge and experience. A couple more quick slides here. Just didn't really talk much about genetic testing, other than to say in the privacy and informed consent context from one of the earlier slides. But there's a lot here just in terms of genetic testing, and that would be for any of us, for example, to give up a genetic. This is what you do for 23andMe or for Ancestry.com, but there are other companies out there that look for disease, specifically for disease markers. So you can imagine if anybody's ever had a claim denied by an insurance company or a failure for them to insure you at all. There is a huge potential for us to have our future health impacted by the misuse of our genetic information. So this could be 10 other talks, but just a few of the things that come up, even when, for example, parents are looking for a genetic diagnosis for their child that they know just isn't right, and it's perplexing their pediatrician, and there's just a real long diagnostic journey. What if you find something in the pursuit of that immediate search for a cause for the symptoms that will demonstrates a disease that will emerge later and maybe in adulthood, and what do you do about that? About 20% of genetic tests on children in similar situations come up with surprising results relating to paternity. How do you deal with that? It's just like some very fundamental, but also some very sort of weightier, more philosophical results that can happen from genetic testing, that I think as a society we haven't really mulled over and really considered with much great depth. So I just wanted to throw another just specific element in there to talk about the complexity here. And then finally, again, back to the second slide I showed, really, there are lots of considerations. Mostly what I focused on were related to the use of these interventions therapeutically and in research. But we've got there's just a whole universe of considerations that we haven't done that well with historically as a society. 100 years ago, in the 1920s, there was, the Illuminati were really fascinated with this concept of eugenics, and that came from an application of a very rudimentary understanding at the time of Mendelian genetics before Watson and Crick had their really illuminated the DNA molecule and how proteins function, or proteins were produced based on that, and the complexity of genetic inheritance versus the more simplified view that was happening in the 100 years ago. So again, lots to consider here, lots to talk about, which I look forward to doing in this section. So hopefully that gave everybody a reasonable, fundamental understanding of the state of the science, at least therapeutically, and then hopefully I didn't scare you too much. So that's happy to take your questions and comments and criticisms. Hopefully the latter, there are fewer than questions. So
Rafa, thank you so much for a thorough presentation. You condensed a very deep topic into an understandable format. I know we have many questions. I'll start with one that came in during the talk from Val. I'll read it from the chat. How is the human DNA in AAV treatment duplicated or propagated in the body, or do you have to continuously supply more DNA in virin capsules?
No, it's a good question. No, it's only given once. So once the virus infects the proper tissue, so say it's a portion of the brain or it's the liver, and the selection of the virus. Some viruses have a greater propensity, or what's called a tropism, for one tissue or another. So if it's a muscle disease, you might choose an AAV that goes to the muscle. If it's a brain disease, you might choose an AAV that selects better for the brain. But you only give these once they get into the nucleus of the, the many brain cells, those will start producing the protein relatively indefinitely, depending on the tissue. But that's one of the things that we'd love to have the time machine for. How long? So in sickle cell and in hemophilia and some of these conditions, they've been working pretty well for years, but we just don't know. But you can only give these once.
Okay, we're going to stay in our Zoom audience for more questions. It says that Al Christians has questions. Al, ask your best one, and we'll come back to others if we have time.
Okay. You've outlined this ethical framework in which you operate, but I know that ethical frameworks only go so far, and the alternative to ethical frameworks is that there could be some things that are just so scary we shouldn't go there rather than to use them ethically. Are there things in this field that you think maybe we won't be able to control if ways are found to do them? What are the things that scare you that might produce strong incentives to ignore ethics?
I'm so glad you asked that question because it's really important, and I'm petrified. So it actually, Al, has already happened. So folks may have heard about a case. I think it was from 2019 where a Chinese investigator inserted a gene into twin girl, and they were embryos. But he inserted a gene into these two embryos that is protective against the transmission of HIV. So he was smart enough to say, okay, I'm going to put in an enhanced, basically an enhanced protection for these two girls. But he put that into their genomes, and they're going to pass that down forever. So that we don't know what could happen down the line. That could turn on other genes that are harmful or detrimental, and whose consent did he get to do that? They're within the community that does genetics research. There has been, a really hard wall or a hard ceiling on this kind of what we call, heritable genetic interventions. And so, but the technology is there, and people have to choose to obviously adhere to these prohibitions and these and these sort of standards and guidelines. They're really not codified into laws. And so the question becomes, how realistic is it to assume that everybody's going to stay on the honor system? I think that's a very scary that's something that scares me a great deal, because the technology is there and people will misuse it. We have a history as humans of misusing technologies, or of exhausting natural, not to be too pessimistic, of exhausting natural resources, or if there's a financial incentive or another incentive to these outside forces to influence the misuse of these technologies, just like others, it's there. So completely agree with you. That particular example is what we point to most often. It scares me a lot. There are other, bioweapons indications here. There are all kinds of, I choose to be an optimist, but there are many reasons to be concerned.
Let's go to the friendly house for a question.
You mentioned the current lack of being able to re-administer doses. Presumably, nanoparticle delivery would be less susceptible to that than AAV delivery. And so I don't know whether any of the current therapies have a nanoparticle component that could be administered after AAV, should it be necessary.
Yeah, there's one. So the only CRISPR program that was approved earlier this year is delivered by what they call an LNP, a lipid nanoparticle. So just as the science is advancing quickly, that's a hope that that with the LNPs or other methods that we'll be able to still be able to hide the DNA that's in there from the immune system, and limit the limit its exposure so that it doesn't target those in the future. There are also various methods to try and mitigate this, but as an ethicist, I'm like, those need to be front and center too, because some patients do show the need to be redosed, and those few that I showed you, and there are many others, given that big funnel of gene therapies that are being developed now. Many patients on any given day are unfortunately underdosed or dosed at the wrong time. So we really want to limit those, and redosing would go a long way to doing that. So yeah, hopeful for the future on new delivery modalities.
Let's go back to our Zoom audience for a question from Hank Robb.
Hi. Thank you so much for the presentation. I want to go back to the history a little bit, because you said that Mendel's work was lost, which I'm sure wasn't the case since we have his notebooks. So what happened was people didn't pay attention.
That's
what
right.
was it all about?
Yeah, I mean, it was published actually, and for whatever reason, I don't know that it'd be a great. I'm not a professional medical historian, but it was actually published. But for whatever reason, I don't know if it was in an obscure journal or what, but it wasn't. That publication was not rediscovered until about 35 years after he published it. So it's really unfortunate. Hard to know where, it's sort of be like a Philip K. Dick novel about what's the alternative future if we'd had those 35 years back where the information wasn't lost.
So you don't think there were some social political reasons for don't look over there, look over here?
Good question. I don't know. It's a good question. Yeah, but as you point out, like those forces can can influence. Well, maybe that's a bad reason to ban books, for example. I wish I had a clearer answer for you. I'm not sure why it was lost for so long.
Thank you so much.
Yeah.
I'll sneak a question in here before we go back to the friendly house. You mentioned the high cost of genetic therapy right now, and we've seen in society that costs of technologies, whether it be computer or medical or other technologies, tend to come down over time. What are some of the key things that need to happen in order to reduce the costs and make it more available to the mainstream?
Yeah, it's a great question. So just to give everybody some context, these gene therapy interventions, again, they're ostensibly supposed to be given once. So they cost anywhere from $2 to $4 million, single dose. So that's a lot of money. And there's no way that, again, developing world healthcare systems can afford that. So the ways that the cost can come down is first of all, the cost of production. Just to engineer enough of those viral particles just in an AAV context is just it's so expensive. Unfortunately, a lot of these a lot of the dollars and cents are sort of well-kept industry secrets, how much their secret sauce costs, but the cost of production and the time for production is absolutely way more than if you were making an organic chemical that's turned into a drug, what we call a small molecule drug. Those cost pennies per dose to produce. These are more like hundreds of thousands of dollars to produce a single dose. So the cost is enormous. The other element is too that again, these single gene mutation conditions, the vast majority of them are rare, and so you have a very small market. And so the company, I'm not justifying this, I'm not on the commercial side of the house. I think everybody should get access to everything, but that's not how the economics of this system are built, for better, for worse, I think. But because there's such a small market, the prices are very high. So they really try to recoup and profit from recoup investment and then ultimately have a sustainable profit. So I think the real economies of scale come with reducing the costs of production and potentially applying the technology across broad scales of rare conditions. So companies would have a whole portfolio of gene therapies, for example, across many diseases that they could ultimately reap a reasonable recoup of their investment.
Thank you. Let's go to question number 2 at the Friendly House.
Yes, thank you, Rafael, for your great overview. For me, it blends into the ethics of human reproduction, like in your example of the Chinese scientist who injected the HIV resistance gene into the 2 embryos. And then, of course, those embryos have become humans by now. And I think about a friend in Ghana who did some genetic testing and found she was a carrier for sickle cell anemia. And the person she decided to have sex with lied to her and told her he had no genetic disposition for sickle cell anemia. Turned out he had full-blown sickle cell anemia, but hid it well. So both alleles with sickle cell anemia. And so I'm wondering if in the future we'll have ID cards that say what our genetic makeup is, kind of like when we travel internationally and we have the injection cards.
Yeah, I sat on a panel last year at the big American bioethics meeting, and there was a medical geneticist from I'll say a nameless country in the Persian Gulf region, and as many know, I think in some in that region, in some of those cultures, there is a lot of what we call consanguineous marriage and reproduction, and that's like from first cousin. That's kind of where we draw the line here in the United States. And he mentioned that they were, I can't remember exactly how he described it, but he said that there was a national program for genetic testing in their country, and they were prohibiting marriages. He was talking about the social implications of actually the government prohibiting marriages because of or reproduction because of the results of genetic tests. And so it's an interesting, again, I think novels have probably already been written, movies, screenplays have been written about what a society like that would look like. It's kind of a blurry line between that and eugenics, but it's an important question. I'll just throw one more anecdote in here because it's so critical and so similar to the story you told. A story of a person, a single woman, and that I've worked with in the past as an advocate, and single mom, wanted to have a child, couldn't find the right partner. Went to a sperm bank, didn't realize she was a carrier herself of a certain disease, not sickle cell, and chose a donor profile and was inseminated with that. And turns out that her daughter has a rare devastating neurological condition similar to Tay-Sachs disease. And so that was just an incredibly unlucky draw from the genetic lottery. And so if everyone had their genome sequenced and had their carrier status for any particular genetic disease identified, and you chose your partner according to not having that status, that would be an interesting utopian or dystopian society. Not sure.
Let's go back to our Zoom audience for a question from Helen.
Hi, wonderful presentation. Thank you. I really understand some things, complicated things now that I didn't before. My quick question, the sickle cell, was that a dose that all those people received, or did you have to make a specific medication for each individual? And the same with the neuromuscular disease?
Yeah, it was a single, it was a single dose. They got lucky. Everybody got the same dose. So what you do in that, and that's a little bit different method, but they got lucky with their first dose. And then in the SMA study, they tried their best. No one wants to start too low,
Right.
but it's a balance between, ooh, if we start a little higher, will we harm them because we're giving them so much virus, or we don't exactly know. It's all based on usually on animal work. But from gene therapy perspective, I believe we have to be better about where we start. And so the sickle cell, that's the reason I put that up there. The sickle cell program got lucky with their first dose. Everybody benefited. The SMA gene therapy started too low, but they got it right on their second dose. Not everyone gets it right even on their second dose, unfortunately.
But they didn't have to everybody got the same medicine. I mean, that's pretty amazing. That's amazing.
Transformative, yes.
Before I go back to the friendly house for a question, I'll read a quick question from Gretchen. I think you answered it a little bit in Al's question, but you can elaborate if you'd like. Is the transplanted genetic material inherited?
No, none of the ones that I showed and none of the ones that are FDA approved are. But in the question in Al's question and then in the other question, that is so the HIV, the anti-HIV gene that was inserted into those embryos, those twin embryos in China by Dr. He, He. You can Google that. That is heritable, and so those twin girls, should they have descendants, all of them now have that in their genome, for better, for worse.
Let's go to question number 3 at the friendly house.
I am thinking about the gene therapy, although it's not called gene therapy, for crops, to change characteristics of their crops so they are resistant to disease and so forth. Of course, there's a lot of resistance to that, and you can see food products that say non-GMO, like GMO is dangerous. But anyway, the genetic engineering or the genetic modification that's been going on for plant crops, I think it's basically the same in principle, or although not in the details, of course, as the gene therapy that you're talking about. So have you and other people researching gene therapy for human health learned from the genetic engineering that's been done for crops?
Methods are similar, not the same, but yes, I think the plant work came before the human work, and because it ostensibly has, although I could get another ethicist beside me to say that the implications are just as important, and I think they are. I think if you look at any situation where you're introducing, a non-native species into, say, into the flora of a forest, or into the sea, into the marine, so there are all sorts of implications to that. I know that in the plant world, in the genetically modified crops world, there are simple questions about, well, we've genetically modified these crops for higher yield. We've genetically modified these crops to be resistant to various pests. What if the pests are going to continue to thrive themselves? Are we driving them down a different evolutionary path to look for a different food source that has a different environmental impact? So these are all really important questions that are more macro. I think within within the human gene editing and gene therapy context, it's been very focused on curative interventions for a particular genetic condition, rather than enhancement, or rather than and with the hard wall against modifying heritable parts of the genome. That those have been the guardrails. So it's a little bit the possibility for proliferation of unintended consequences is sort of maybe we're being sanguine to think this, but we think it's a little bit lower. But it's a good question. It's an important question, and one that I think is is very relevant.
Let's stay at the friendly house for question number 4.
Okay, I guess that's me. I just yesterday received a test kit from OHSU for a cancer study where I will be spitting into a tube, and as well as answering lots of questions on questionnaires and things. Before I do that spitting, should I worry about what the potential repercussions down the line from having my DNA in this study should be?
The consent form should very clearly identify what they intend to do with that sample. So I would, hopefully it's not 30 pages long, which is another set of problems. But the mitigation for that concern, which is a very, very valid concern. Hopefully I didn't make people more scared about it today. Is informing you of what the genetic information will be used for, and what the limitations of that are, that they won't sell it to anyone, that it will go no further, that it will be used only to either develop a potential treatment for you or for others with a similar condition.
And I must admit, I'm a little surprised that they would even find this valuable. It seems like there's so much data available now to these people, that having this additional volunteer come in and provide this information, it surprises me a little bit.
But it's all about every additional sample is often useful. I mean, I think if you think about, in Iceland, the entire population has their genomes sequenced in their government database, and similarly, the UK has what they call the UK Biobank, and those are interrogating those genetic databases has given some really interesting information. I'm not a bioethicist that focuses on informatics per se, but like those implications are also enormous and can be really useful.
Before we get to question number 5 at the friendly house, I have a second question. That's the benefit of being an MC. You get to ask 2 questions. Keep that in mind, people. Okay, it seems to me that we have a huge divide, especially in this country, between the science and the anti-science populations. We saw that with the COVID vaccine. Is there any talk in your field that we are just too divided right now, or too scientifically ignorant to advance this field much further?
Yeah, that's a good question. I guess the short answer is no, not really. I think the focus, as I mentioned, is so much on treating children with these debilitating conditions, or adults that have suffered a lot. And so we really focus on keeping things into that context. And so most of the time, parents will do pretty much anything for their suffering or sick child. Similarly, if an intervention like this can have a major impact on your quality of life, reduce pain, etc., then it's not a tremendously complicated decision. But as it branches out into further, like aging is the one that I actually worry the most about, because first of all, it's so multifactorial, but But secondly, it's just such a huge business, and there are so many people that do not embrace the things that happen with age. I being, one of the certain things like my reading glasses. So it's just it's the broader use of the technologies beyond the solving some of these more discrete issues that's a little troubling.
Thank you. Let's go to question number 5 at the Friendly House.
Yes, I had before I get to my question, I had a small correction to make. The Chinese case of editing of the CCR1 gene was not insertion of genetic material. It was using CRISPR-Cas9 to make small deletions in the gene, which was an attempt to mimic some naturally occurring mutations that exist, but doesn't actually mimic them. So the second one was my second point was that in the this issue of reproduction and being able to reproduce more safely if you're a carrier of a genetic disease, I believe around 2000, the year 2000, that, and I think it was Lee Silver who tried to form a company to basically genome sequence sperm donors so that they could do exactly that. And for some reason, that company failed, and I'm not sure exactly why. It might have been that sequencing was too expensive at the time. But now, with whole genome sequencing being like $500 for a whole genome, it really seems like that would be a very reasonable thing, if not really an ethically appropriate thing to do, is to insist that all sperm donor banks actually sequence all of their donors to find recessive lethal mutations.
It's a pragmatic, today I think I don't know what the costs were back in the early 2000s, but they were probably orders of magnitude more than they are now. So that may have been a financial reason why it didn't make it. And as you point out, for whole genome or whole exome sequencing, the costs have come down further and further and further. They're still not cheap. They're still unavailable to some families. But I take your point. I think it's solving one problem often creates another, and not to be dystopian here, but again, anyone in the sperm banking business or the reproduction business would also need to obviously ensure privacy of that genetic information for any of the sperm donors. So it's but yeah, to your point, I think it's I would want to know. I myself would want to know. Others, however, are much more willing to roll the dice. And so, one of the big ethical discussions is this concept of of right of an open future. And so the example that's used is that you test a child for you're in search of a genetic disease that they're showing symptoms for in childhood. You think it might be something, you send out a genetic test for that, but no, it comes back as it's something else that's not going to emerge. It's going to be lethal when they're 40, say Huntington's chorea. And what do you do? So it's not what you were looking for. You find something else. What do you do? When do you tell that child, or do you give that child the option to want to know sooner or later what their future is? Some people answer that question, I don't want to know, I don't ever want to know. Some people say, you must tell the child. Again, there's no real right or wrong answer there. I know my personal beliefs about that, but I think you point out an interesting question, which is difficult to resolve, that different people have different opinions on how information like that should be communicated and managed.
We have an incredibly smart audience here, both in person and on the Zoom, because prior to that question, Val also wrote, by the way, the Chinese experiment did not introduce a gene resistant to HIV. It disabled a normal gene that HIV needs to infect.
Right. I appreciate the correction. Yes, and it was not a gene replacement. It was an edit, not a. Yeah. So I mean, the point here is that by taking some genes, put the brakes on things, some genes accelerate things. Some genes modify the brakes or the accelerator. So there's a lot of different impacts here. So I appreciate the specificity there. But it was a CRISPR intervention, not a gene replacement.
And Rafael, this was an incredible talk today, and really a spirited Q&A as well. You've clearly spurred a lot of curiosity. And one of the things I like to leave our presentations with is what resources, what mediums do you recommend people can follow to stay abreast of this field?
Yeah, I mean, there are lots. I think, you watch, I track things like, the New England Journal of Medicine. Just, you don't have to subscribe to them, but you can just look at it's a once weekly big medical publication, and they splash all of their big studies on their homepage. Every Thursday is when they release new editions, and a lot of their content for a breaking article that they think is social, where there's a lot of social interest, is free. So that's something that I watch very carefully. I think other areas that I think a great book is called The Gene by Sid Mukherjee, who's at Columbia. He's actually an oncologist. If you have a real interest in genetics and biotechnology and gene therapy. He also wrote another famous book called The Emperor of All Maladies, which is about cancer, about 10 years ago. So I really recommend his book if you want to sort of increase your literacy about genetic biotechnology. But those are just in New York Times. Their science section really follows. Gene therapy is of interest to people now. So there are plenty of misuses of genetic type therapy. People talk about stem cells. People talk there's just whole areas that have to be regulated that aren't quite regulated yet, and so it's a very dynamic environment at the moment, but yeah, or just email me. I'm happy to entertain questions. I really focus on research ethics and pediatric research ethics in particular for these advanced therapies. So I'm busy, let's just say.
Rafael, this has been one of the best science talks we've had in a long time. Thank you so much for your presentation and for your time today.
No, thank you very much. Really glad to speak with you, and let's stay in touch, okay?
Appreciate it. Thank you.
Okay.
Take care.
Ciao.